I am KK Lamb. I am the project director for the newborn screening ERG. I want to give thanks for the invitation to speak to this group about evidence-based reviews for newborn screening, a closer look at the review process. I wish I could be there in person, but I'm really thrilled to be a part of this boot camp again. It's an important piece of the newborn screening. I've been the project director for the ERG since 2011, and I've had the honor of working with Dr. Alex Kemper, who is the lead, and we have been funded through HRSA contracts continuously for this work, and therefore the disclaimer here that this particular piece here is not part of those contracts, and sure that they are not the views of disclaimer. These are my views and my work, and though of course was reviewed by HRSA. So today, again, I'm going to be just giving an overview basically about how we do things, closer look at the evidence review process. Briefly, I'll just remind us of where this evidence reviews for newborn screening sits in context with the committee's process, and then we'll go into the methods for these reviews, conceptual framework, over mission, data sources, how we review them, key topics, and guiding questions. A little dry of a talk, but hopefully I'm going to stay awake. First of all, evidence-based reviews and context for newborn screening. Of course, the full evidence-based review doesn't occur until and unless the committee requests that, and that might happen after a nomination is reviewed and presented to the committee, and the committee votes to recommend that the nomination be moved forward to the full evidence review. So of course, not every nomination goes straight to full evidence review. That gives us the advantage at that time, once the review starts, when it starts on that day, when it's forwarded over to us, we have the advantage, we're able to review and look at the nomination package materials, and also the nomination prioritization work group presentation, and that is all really helpful to give us a lay of the land. We do the full review procedures for every condition, but that way we have a heads up about what issues to be on the lookout for. So where this all started, there's a little fun fact in here. The advisory committee methods development, are looking closely, you'll see that the author here is Dr. Ned Galange, who we now have as the esteemed chair of the advisory committee itself. So this was during his earlier stint, he's mentioned that, and he was instrumental in developing the methods because he was also on the Preventive Services Task Force Committee, a similar making public health or making clinical guideline recommendations based on evidence. Yes, that full reference, that was from 2010. Again, the methods that we use are systematic. They're systematic evidence review methods, and that's pretty specific, being they're pretty standard, though there are some adaptations. So in the 2010 paper, the group presented an analytic framework that draws from the Preventive Services Task Force, And this is here to depict the evidence review and to guide the evidence review for newborn screening, with the overarching, right, the number one line, right, basically the main question being, does newborn screening lead to better health outcomes compared to usual case detection? That's the question at hand. Now, again, as I mentioned, right, the landscape of newborn screening and rare diseases is different. The evidence is quite limited for rare diseases, just by the number of patients affected. The newborn screening at the time of these reviews is not widely implemented. It's not on the RUSP. In some cases, there are no states fully screening. They might just be piloting or something. So related to that, there's little to no direct evidence about screening. No randomized clinical trials, which is considered the gold standard for reviews and scientific rigor generally. So I put in there, down there in the blue, in reality, then the question ends up in what we're really looking for and able to find, what we do find in that evidence base, is that we can look for what is the evidence about the net benefit of early detection. And then it is slightly different as we work towards it. What sources of evidence do we review? These, we go through these in each of the reviews. These kind of standard databases appropriate for newborn screening, PubMed, MBase, Cochrane Reviews. We also do nursing and allied health disciplines, and any others that might be indicated for any given condition or change in time. We do the searches for prescribed period, publication dates, depending on the exact condition. They may be slightly different, but the important piece is that we also have a cutoff of the last day that we will accept new publications. And we have in the past gotten a slew of late breakers. So we just need to be able to have enough time to adequately review them. Another piece that we have is we do review now gray literature, and we review it more thoroughly or more fully now here with the most recent revisions to the committee process. So that might include committee or meeting presentations for all of these. If there is not sufficient evidence to assess the study itself and what they did, we do need to get additional information from the authors about the presentation, like either the presentation itself or whatnot. We also expanded our inclusion and review of unpublished evidence. We work with screening programs, we get their surveys, we do we pull from registries when those are available, that can be published or unpublished. We look at conference proceedings, also not published in the databases. I think the symposium may have some in there, but definitely some of the other condition-specific ones are not in the databases, but have some important information or useful information. We look at clinicaltrials.gov to be aware of emerging treatments. And then the other big piece is that we officially no longer do primary data collection or analyses. At one point we did do those, but we no longer have the time the time to do that given the 9-month time frame, and that these reviews have expanded over time. How do we review the evidence? the procedures follow more closely the standardized methods established for systematic evidence reviews, like used by Cochrane and just the evidence-based review groups that are out there. It's a multi-stage screening process. We have 2 reviewers who go through them at each process independently, and then we confer on any conflicts. We have a set criteria for inclusion and exclusion that are developed from the key questions for that particular condition. Go a little more about the key questions later on, and then also other specified criteria that are more general, like English language, human studies, etc. We also do, for the key studies, usually treatment and screening, we do assessments of quality and risk of bias. These are standardized or established measures, questionnaires used in systematic evidence reviews, and those are all usually found in the appendices, and we also describe give the assessment of quality ratings in the text in our reports. We follow the PRISMA, our preferred reporting items for systematic reviews and meta-analyses, and present that in the diagram. And that's really being fully transparent and documenting everything we've done, the number of articles we got in from each of the databases, and then after each level of screening, we show how many were included and how many then got excluded, and so forth. And the point of all of this really is to be truly transparent and to create objective reviews that could theoretically be replicated by someone, given the dates that we report, that we use, the criteria, pretty specific criteria for inclusion and exclusion. And that helps ensure that this is objective and that the review itself is has a higher certainty of findings. Overall, like I said, our newborn screening reviews have also expanded. So while initially they started off with just this first question, looking at the clinical effects of early detection and treatment for the condition, focusing primarily on the infant outcomes and family outcomes. As I mentioned, the public health impact piece was added on, and I think like in 2013 or 14, that was through mandate. And we also look at population level benefits, projecting the number of infants detected in the population, and different outcomes as we're able to with the research. And then also the readiness and feasibility, basically the capacity of the state newborn screening programs and labs to be able to expand again. Key topic areas. This just shows, going back to the basic analytic framework, and again, that was presented and set forth early on. There are some general topics represented by in the points of the diagram, which is just kind of the flow of screening. And with the overarching one there at number 1, the net benefit of early detection, and then the other topic areas being thinking of the general population, kind of what's kind of the current standard of care, what's the natural history, what's the incidence and prevalence, but then going on to screening, short-term follow-up and diagnosis, treatment, harms and benefits of screening and of treatment and follow-up, the different levels of outcomes, and the more recently added public health impact piece. Now, within each of those key topic areas, We have a number of key questions to drill down about those topics. And we have for each key topic area, we have a number of key questions that are pretty standard that we're always looking for. For instance, epidemiology, natural history, prevalence. We are looking to make sure we understand the case definition and have that clear. Incidence, prevalence of the disorder, clinical detection, and with screening, if there's any information available, the course of the disease, what major systems might be affected, ages of onset, the timepiece is important, ages of onset, diagnosis, treatment, outcomes, survival rates, etc. I'm not going to go through all of these, but that's just to say there are a number of key questions. If you want to go see the questions laid out further, I'm going to be providing a list of different resources to go to at the end of this. One, I do want to talk about just a little bit further to give some examples, right? So when I've talked about looking for, having to look for indirect evidence, right? Because there's very little direct evidence about the benefits of newborn screening. So we gather information about early detection and treatment, and some ways that we can look at indirect evidence or potentially proxies or something are, and these are some examples from completed reviews, or to look at treatment outcomes when initiated early with some patients versus later. So like less than 6 months initiation versus later initiation. Those have been from registry databases, published, unpublished, case series reports, treatment outcomes for patients with shorter disease versus longer disease duration. That was an SMA from a phase 2 clinical trial. Actually, that might be a phase 3 of the entire study, so I apologize for that. The sibling case reports, a very specific example of early detection and treatment initiation. And those can be really helpful if there are kind of a number of like 1 or 2 case reports may not, may be helpful, but not kind of like really start to strengthen the evidence. But if there are, I think we had like 6 or 8 in the last couple of case reports. And so we were able to start to see some trends and show some of that. These case reports, particularly would need to have like timing, ages of onset of symptoms, age of treatment initiation, ages at different follow-up periods, outcomes at different follow-up periods, and preferably consistent, somewhat comparable measures across the two. So those can actually be pretty powerful, and something that patients and families in particular are needed to help report these. So summary and conclusions about evidence-based reviews and our methods. They're based on standardized methods from the task force, but are adapted for rare diseases and an intervention that is not widely implemented. So there's little direct evidence. It's inclusive. We are more inclusive of data sources than any other standard or systematic evidence-based reviews. We review gray literature, conference abstracts, unpublished evidence, as long as they meet some very minimum criteria that we can at least assess the risk of bias, then we can include them since they continue to be relevant to the key questions Hopefully you can gather also that it's really actually, for ours, it's important that we have information and evidence from families and clinicians from a wide range, families, clinicians, researchers, industry, public health labs, and others. There are many dimensions to newborn screening, so we need all of those views. And lastly, the resources page I just wanted to provide, since there's a lot more detail out there. One is the committee website, newly updated. Generally, there's also a new section I want to highlight: condition, nomination, review. That's where a lot of these materials came from. In the report section, you can find two of the major methods, review process updates. Under the previously nominated conditions, you'll find the previous evidence-based review final reports and the methods and appendices. Outline a lot of the methods, and then if you have any other questions, feel free to contact me. Thank you.
Hi everyone, I'm Terry Klein, and I am the President and CEO of the National MPS Society. And first and foremost, besides that, I'm a mother of 4 children, and our youngest daughter, Jennifer Klein, has mucolipidosis, and so she was my kind of like our point of entry as a family when we were diagnosed with this disorder 22 years ago. My experience in newborn screening really stems from my predecessor, Barbara Wiedehase, who had put forth the nomination with the expert committee for MPS 1 back in 2012, and then watching that unfold, and then of course the most recent MPS 2 nomination that went through the process with the ACHDNC, and then it was recently added to the ROSP.
Great, thank you. Is this still, okay. I think it's on, yeah? Is this better? That's okay. Should I switch out with that one? Great. One moment.
Technology. It's great though. Okay.
Oh, yeah. Ooh. No, you really can hear me. Great. So kind of to kick off this conversation, you know, KK just walked us through the evidence review group's perspective on the review process, all the different pieces that go into that, all the things they think of. And, you know, having gone through the review process yourself, you know, what's your experience as a patient advocacy organization? You know, the other side of all of those flow charts and pieces that were just presented.
Yeah, KK did a really great job of showing you guys the grids, and one of those flow charts was a little small, but this information is available on the HRSA website. I brought my computer. If anybody knows me that's known me for a long time, I don't technically look at my notes. I try to remember everything, but this is just so important that I didn't want to skip anything. And so I have some things written down on my computer. I apologize if I look that way. But I want to first share with you on the experiences that I'm going to primarily rest in MPS 2, but recognize this is the second time the National MPS Society has gone through this space. The first time we went through was a decade ago, and the nomination for MPS 1 was successfully added in 2016. And the process was a little different, and it was a nail biter. I will say this, with the MPS 1 nomination process through the evidence review, we had 2 considerations for treatment at that time, and that was because MPS 1 was, we had the bone marrow transplant, which was available, and then we also had available enzyme replacement therapy. And so we had 2 different therapeutic approaches that were being measured. And it was really a nail biter, and it was quite different. But let's kind of stay within this MPS 2 RUS nomination process that we recently went through. Overall, I think I have to give credit to a few places. The first of all, it was representatives, clinical team from Takeda, who really helped us get this started to begin with. So when someone here had asked the question, who submits, right? Who does the submission? This was so important to know, because in this case, we had industry that came to us and said, for us as a patient advocacy organization, it's not always about the money. Sometimes it's about the resources and the knowledge. And if they can at least spark us to get started and plant the seed, then we're going to be able to take that nomination and go forward. And that's exactly what they did. I will forever be grateful to Katia Wright and what she did to help us bring this nomination forward. Also, we have to give credit to Dr. Matthew Ellenwood, who isn't here with us today, but he joined probably at the fourth quarter, almost at the Hail Mary point in terms of the nomination going in. We had to do some rewrites with it, and he joined the National MPS Society during the pandemic when this nomination had already been presented. So let me just kind of rest it there. Overall, we thought the process went really well. We were quite pleased with the amount of communication that the evidence review group provided to the National MPS Society. A couple of the areas, though, that we had concerns, and I want you to know that that part was the positives, but I always want to point out the things that I felt were challenging that I think that we can do more with there were a couple of metrics that we knew were going to be measurements during the evidence review group that had to do with the Office of Management and Budget at the state levels for the state health labs, as well as the surveys that they're going to be using to see how well they're performing the function of the screening. And those were things that were kind of out of our control. We knew that they needed to be measured. We knew that those were going to be part of the entire matrix overall, but we really didn't have any footing in that space. And so when we were going through the evidence review for the MPS 2 nomination itself, we recognized that when we brought this, what they did was we come together on Zoom, but we had a group of statisticians, a wonderful group from I think the University of Michigan, who was there to help us understand what these decision trees were and what these definitions were from the surveys from the state health labs. And they were a little confusing. They were a little clumsy. We definitely had our research committee, our core technical committee of Dr. Burton and Dr. Munzer and us from the National MPS Society kind of questioning, can you tell us what that means? I'm not quite understanding that. We knew that they understood it, but we couldn't understand it as a patient advocacy group. But we knew it was a critical part of the component of how we're going to screen and if this nomination is going to go through. I just wanted everyone to recognize that there is this piece, and it's certainly outlined. It's definitely outlined when you're submitting your nomination, but until you're in it, I don't remember that type of a feedback being so much of an issue with the MPS 1 nomination as it was with the MPS 2, and we were really dissecting that because in the end, you're going to be graded for these areas. I wanted to say also that we were grateful to Dr. Alex Kemper and to KK and to the group because we had a number of questions, and at times they were just simply naive questions of things we didn't know. And we found that the responsiveness from the team was really quick. We were not waiting weeks at all. You were getting responses in the same day usually, if not the next day. And then I wanted to talk about the importance of what we called, twice now people have brought up, called the gray literature, and that evidence review of the gray literature, because we did want to put forth some things that were gray literature and what would those look like. And those happened to center around things that were data that we had from Missouri, some state health data that needed to be looked at again and analyzed and verified. We had some data from babies that we wanted to take a look at and have it potentially submitted. And so we were a proponent of potentially this information. They also talked about, and I think KK had one of the screens up there about conferences. I'm not sure what she meant by the conference piece, but what I wanted to share was it provided us a time during the evidence review group to hear from the patient advocacy that we know 20 years ago, we did not have adults living with Hunter syndrome attending the conferences. 20 years ago, when I went to a conference, it was more of a pediatric type conference, unless you were very attenuated with the disease. And ERT has changed everything. And I wanted them to know our patient community is quite vast and quite large because of enzyme replacement therapy. And now our conferences are filled with adults that have had these life-saving therapies. But just knowing that that could be a report or a part of the gray matter, the gray literature, I'm sorry, that you're going to add becomes very important. The other part I wanted to pull out for this question was around how articulate and clear that the evidence review group was to us about our gap in information. They were able to really point out to us what they thought still might be a missing component. And even though that those were hard to hear, you don't know what you don't know. The original nomination went in with 37 or 39 cited resources, and it had a wonderful committee that wrote this nomination. But once you have your piece together, pulling out your gaps is very, very important. And these were, so I'll say them really quickly. We were short on sibling studies, and we heard that from KK. She brought that up. The sibling studies, how important they were, and how hard it is to have sibling studies from a rare disease community. How can you have that evidence? The other area was longitudinal studies across the enzyme replacement therapy. They were looking for more evidence. And so hearing that information, and if you remember KK's screen, she had tiered out for you peer-reviewed evidence, gray literature evidence, and I can't remember what the third one was, but there's a third tier there in terms of what unpublished data, that's what it was. We knew though, as a patient advocacy group, that if we could get the unmet need and the gaps to published or peer-reviewed levels quickly, that this was going to help our nomination. But here was the challenge. We only had less than 4 months to do it. You see, we're in the middle of the evidence review group meeting for 9 months trying to gather this information, and we had 4 months to do this, and so we did. And so through the efforts of Dr. Ellen Wood, who managed this process for us, project managed the process, we were able to put forth a sibling study from Dr. Julie Isengart and Nathan Grant on an MPS II boys in a family who were treated at very different points of their lives with ERT, and one is now moved into a clinical trial for another second generational drug. It's a fantastic paper. There was a sibling study done by Dr. Linda Polgreen, who looked at some longitudinal evidence of the effects of the ERT. And then we also had a wonderful publication done by Dr. Barbara Burton and Dr. Joseph Munzer from the Hunter Outcome Survey study, looking at longitudinal treatment for these boys with Hunter syndrome. In having those land either in peer-reviewed publications, Linda Polgreen's went to a world abstract, became pieces that the ERG allowed us to submit with our nomination. You see, this was still open. And so if you can do this, you can get those pieces through. That's what's really important. And the lessons that we learned here from the MPS 2 versus 1 is how to have just as responsive people as the evidence review group from the ACHDNC was to us, how responsive we needed our team to be to jump all over these items. Because again, we had 4 months to do this. And we know that it made a difference.
Great, thank you so much. I think that's really, it's just so helpful to hear the real lived experience of going through it. I realized that I didn't introduce myself. I promise I didn't just stroll off the street and say, hey, this seems cool. Let me see if I can contribute anything here. I know, thanks. I'm Natasha Bonham, founder of Expecting Health and director of Baby's First Test. And as part of those roles, I have been to, I think, every single Secretary's Advisory Committee meeting since 2008, minus 2. And so in that, and being an organizational rep and being a non-voting member, but seeing families come and nomination packets get submitted and reading all of that information, I think exactly what you said is right in terms of it is evolving, right? It's changing and we're learning more, and the Evidence Review Committee has really worked to keep up with the pace and to get that feedback back. I think it was the last Newborn Screening Saves Lives Act put into effect the 9-month turnaround time. So what that means is from the time a nomination is submitted, there's 9 months to review it and for it to get a response back. And that's great, right? We want things to keep going, but then that means there's 4 months for that evidence review and the back and forth. So just trying to give some of that context in terms of that timeline and why it can seem like a rush to get information back. One thing I wanted to ask is, now that now that this data has been collected for the evidence review and gone through that process and you're on RUSP, what information do communities need to collect once they are on the RUSP and various state panels? So, everything's gone well, right? And what I mean by that is nothing is linear in life, right? Including this. There's conversation, there's back and forth, there's sometimes conditions get sent back and you have to go back and resubmit. That is all very normal. But then you're added onto RUSP, you're getting onto the state panels. Data doesn't just end there, right? What data should communities be thinking about from that viewpoint?
Yeah, it's really just the beginning. Of course, we have to give credit to the decades of work from the researchers and the scientists before this nomination went forward. Because without, I said, those 37 to 39 citations, we wouldn't have been able to put the nomination in front of us. But it doesn't mean it stops there. It's been added. It's about a program that is already successful. Newborn screening is successful. It's a successful health program. But there's data that can be drawn that can make the path better as we move forward. And if we don't take those opportunities to get that data and recognize where that data can be, and how we can extrapolate it, understand it, quantify it, and be able to make a better path forward for other diseases that may have nominations or be screening, then we're at a loss. And so one of the areas that I thought about, and I haven't even talked to Dr. Matthew Ellenwood, our CSO, to flesh this out some more, was it certainly would be good to be able to collect data, I think, from the state health labs in terms of how their readiness is. How are they measuring their readiness? I still think we walked away with a vulnerability of not quite understanding how the states thoroughly did their surveying for their operational readiness. And what can that data look like? And more importantly, as I think Dean and Sylvia was talking about in the last panel, patient advocacy groups have an opportunity to partner with their state health labs, and they should, because this is listening sessions for both sides. It's vulnerable because we want the baby screened, but if we can partner with them, we may have a better understanding of this important grade that becomes part of the matrix of the decision tree by the ACHDNC. And I just think there's something there that we could be drilling down in better. But I haven't fleshed the idea out, but I didn't want to lose that one. So let me go into a couple of the other things that I have here. We have gone through a number of publications, but one of them we're just an honor to be a part of, and that's through, gonna get out my notes because I don't want to mess it up, I want to say it correctly. It's our MPS GAG biomarker methods, our quantifications, and it's work that we've been doing with Zachary Herbst and Dr. Michael Gelb. And some of the publications that you'll see come from that were examples of evaluations of multiple methods of quantification for glycosimic glycans. So we can take a look at newborn dried blood spots from patients that both had severe in the MPS 1 and attenuated MPS 1. And we're looking at things like the dried blood spots afterwards, looking at the data, quantifying the methods in terms of how that work is being done. And maybe Dr. Gelb will be able to explain a little bit more about that at some point during this day. Those types of areas are important. Some of the other publications for data that we have thought have been very important is Dr. Matthew Ellenwood had just released a new publication around the screening and the RUSP experience. And so understanding how we capture data differently this time through MPS 2 versus MPS 1. And so I would be looking back at that article because we didn't want that to go nowhere. We wanted to be able to put forward for organizations coming forward what 10 years look like of collecting data and information, because we have a decade-long experience of doing that now. The other area that we found to be very important is when we see gap points that we knew we couldn't succeed in. So we walked away from the evidence review group with ideas about where there's gaps for the future that we need to have data. And one of those was around consensus guidelines and care management for these diseases from the physicians. We didn't have real solid piece there possibly, or multiple consensus around Hunter syndrome in the care management. And so as we're looking at the newborn screening program as a whole and holistically, we're screening the child, they're being educated about the disease, where's the consensus in terms of how to treat them, not just with the ART, but after in a lifelong space. And so society walked away from knowing that part of our grant process, our third tier cycle this year for our grants, is $25,000 grants to researchers who will work with us in building these consensus studies and gathering this data. And so we're grateful to going through the ERG because they pointed that out to us, and that was really important. The other area that we have is unique to the society, but I need to share it for data gathering. We have a Pathways program. And our Pathways program is a program where social workers meet face-to-face with our newly diagnosed patients. And we started the program serendipitously in 2017, right after MPS 1 was added. But it wasn't because MPS 1 added, it just serendipitously worked out like this. We had always had a vision if we could go beyond telesupport and telephone support to have social workers meet with our families and licensed social workers that could meet with the families around the country. They could wrap around not just the disease of the family and the diagnosis of the family, but holistically around all things that social workers do so well, which is look at the environment and look at the areas of health inequities and food inequities and all the things that could make life just a very difficult place to be. But because of that, we have data. And from that data that we have, we're certain we'll be able to bring some of that data forward. It's going to go into publication soon, the first round of it. We now have an end of 374 families that have been through the process. It is patient information. They have consented, and we're surveying them across very key diagnostic processes. How was the diagnosis process for you? And I recognize these are with the mucopolysaccharidosis and the families for MPS 1, MPS 2, and our other disorders that don't have newborn screening, but how was it for you? And how can we gather that data and bring it forward, because we know it doesn't exist anywhere else in the world. And so you have a responsibility to bring things back to the newborn screening program that are going to be extremely beneficial for the future.
Great. I really appreciate you talking about this broader data. I think sometimes, especially when we have such a focused goal, we say, what do you want from us? And we'll build towards that. And what you're really talking about is really more overarching, really collecting the data of the experience of families from a multiple different lenses. And then when you have that information, you can then pull that and say, okay, well, for a Russ submission, we have this kind of data, but for our other work, our other organizational and community work, we can pull that information. And I think that's really key, especially right now, as there's more and more of a conversation in the newborn screening community about long-term follow-up. What does that mean? Where does it live? And again, we always say it's not about a test. It's not just a test. So our efforts have to be more than just the test. It needs to say, what's happening with these families 5 years after a diagnosis? And so I think that's great that you've already kind of built that infrastructure with those and pulling that data together. Let's see if there are any questions in the room or from online. There's one right here in front. They're getting you a microphone.
I have a question. When you were talking about, when you were doing the nomination, you were saying that the University of Michigan helped you to go through the process because it was some words that it was difficult to understand. So do you recommend that we find somebody? Do you guys have any information that can help us to go through this process?
Thank you for the question. So I'll frame it for you more. This is a group that's brought forward by the ACHDNC Evidence Review Group. They bring those statisticians into the nomination process. I think some education or some pre-meeting possibly with the patient advocacy group that's going to have that nomination to better understand what's going to be presented to us would be very helpful, because I felt like we probably were doing a little not a push and pull, but we were kind of doing a little push and pull because we didn't quite understand. All of a sudden, we had all of this metrics in front of us from data that we don't have, but it's from the health labs, which is really important. But we were trying to work through to understand what it really meant, what did it look like. That's going to be part of the overall grade for the nomination. So they actually bring the group into the process. And so my hope for the future is that there can just be a smidgen more time spent there for the patient advocacy group to understand that coefficient for the decision process.
And one more question. Is there any information that you can help us or provide us to make it a little bit easier for the ones that we are going to start this process?
That's a great question, because I don't know if everyone knows this. I think Dean and I were touching on this earlier, but one thing we learned from MPS 1 to MPS 2 is that we didn't really take a look back at all the resources that the HRSA site actually has. You can go back and take a look at all of the nominations that were put forward. They have minutes, they even have transcripts for some of them. And to go back and to listen to how they were thinking through the review process, I know can feel arduous, but it was really invaluable for us to go back there and say, hey, did you know this is here? Let's go back and listen to this and see how they got through this. And it also provides you a sense of some comfort, because you have an idea of how they're going to operate, with you. And so I really highly recommend going to the HRSA site.
I just want to add to that. Years ago, probably over a decade ago, our group had a very small amount of funding from HRSA to help in that case. So I'd have patient groups call me and say, we want to submit a nomination, what do I do? And it would be a lot of this conversation in terms of, well, at the last meeting, they were really talking about, the importance of having data from a state and why that was so important, an issue that came up earlier today. And so there was infrastructures were in place, before, but I think especially with how the process has evolved and there's so many different components and conditions are even more complex than they were then, that, it may be something that we can definitely think back, how could we reincorporate? Because sometimes, yes, it's good to go back and read everything, and if you can, that's great, but also to have a conversation with someone. I know people are constantly emailing you and wanting, can I have 5 minutes or 5 hours or 5 days of your time to help me through this? And seeing if there are ways that we can pull our resources together to do that. But it's a great question. There's one.
Yes, hi, I have a question from an online viewer. It is, how long did it take you to put together the overall nomination? And what were the roles of the various stakeholders in putting the package together?
That's a really good question. And I think because of MPS 1, we were able to put the two together a little faster, but we did define roles. And so before Dr. Ellenwood began with the society, I was the project management over this, but really because of Katya Wright from Takeda, who oversaw the medical writing for the nomination of the pieces. And I was just trying to keep things in a project timeline for everyone realistically. 3 years. 3 years was the time that we took to fully feel comfortable in that submission. It was about 2 years, but then we went through the rewrite. One of the words I didn't hear addressed today was the prioritization work group. And that's the work group that actually makes sure that your nomination is ready to go to evidence review. And we heard that part, but that prioritization work group was part. And I'm including that in the 3-year piece. But it took about 2.5 years and then another 6 months under the prior work group to kind of go through the rewrite before it went to the ERG. Everyone had very specific tasks. I know that Dr. Gelb was in that nomination. He was looking at some of the measurements. Dr. Munzer, each of those positions or the specialists were very specific in the area of the nomination that they kind of took responsibility for. And then we all came together on monthly meetings or biweekly meetings, or we would do email exchanges and we would redline and go through things and challenge each other and debate each other. And so they're in that space. But the most important thing I can share with you, and I can't say it enough, is when you choose the committee that's going to be on the nomination with you, and it takes the committee of experts, you want them to be the responsive physicians. You want them to be the ones that will be there to answer those questions quickly and work through those problems quickly, because they're very, very busy. And those, there may have physicians in your disease groups, they'll come forward and say, as much as I want to be, I just don't have the time, maybe I can do just this. And so we did define roles, and then we defined roles even more once Matthew was there, and that's how we did it.
Okay, I have one more question from an online viewer, and it is, following Dean's remarks on the previous panel and the pivotal role the MPS Society played in the nomination, how do newly diagnosed families identified through newborn screening find the MPS Society in a timely way?
Yeah, that's a great question, because Dean brings up a really important, all we're looking for is one line, and it just says mpssociety.org. It doesn't have to say anything else on that sheet of paper. We don't want to clutter it up. We really want them to get soft landing familial pieces at the time of diagnosis from the health lab. But if they could just have that resource on, and I'll be very upfront, a lot of our families come from Google and Facebook. And there's just a really, really popular Facebook, and the families are amazing in that group. It's over 2,800 families now in that group. So Facebook, social media, golly geez, like Dean, I'd like to just get mpssociety.org on the bottom of the sheet.
Yeah, and just to add on to that too, that is such an issue when you're looking at, really almost any disease, but including rare disease. It's something that we've seen so few times that families are referred to support. They may say, oh, you should go find support, and then that's one other thing that a family has to go through. It's one of the reasons why on Baby's First Test, on our condition pages, we have those reviewed both by medical experts as well as the advocacy organization. So then they can tell us which of their websites they want up, and if they want direct resources specifically for those families who are in the early days of a newborn screening diagnosis. But again, and so it's great then when states refer to us, that's wonderful, but we need to get the names and links out of those advocacy organizations closer to that first step, that first point when a family is identified and diagnosed.
Yeah, I think at this point I want to reinforce Terry and I have just this synergy. But it is important that the landing page for those families be for a newly diagnosed page. You don't want them to just come into the main front or, whatever your clinical trial recruiting is. They're long past that, or they may not be. But I want to just, I'm not challenging you, Terry, but I think it's wonderful that you had all the support and have all the support you do for newborn screening, and you have a second condition and you have another one that you're doing and so on. That isn't the situation with a lot of organizations. And so I just want to encourage you that if you don't have a dozen experts and 3 project leaders and 6 clinical trials and 5 pharma companies working with you, that's okay. You can still submit a nomination. It's going to be more work and more focused, but to bite off what you can. Look at the form, go back and look at the previous nominations and responses and so on, and learn from that. We haven't completed the process, Terry has, but having built relationships with some of the reviewers and so on, it's my hope that we get the same interaction and encouragement and could you help us with kind of response when they recognize that we're a smaller group than, for example, the MPS Society. And so I would encourage that back to the rest of you that might say, wow, Terry can just do all these wonderful things, and maybe you're overwhelmed at this point.
No, thank you, Dean, because he makes up a really good point. Here we are, we're in this space, and this is what we do, and we've tagged newborn screening as one of the highest priorities for the society, because for our patient community, this is equitable access for a baby. The babies deserve to have equitable access to health care, and they have the right to have a screening. And so know that one of the things I had learned, because I came from a much smaller organization that was global, do something. Do something, just do something. Just get started. We had to do that for MPS. We had to do that for the Pathways program we started. This is a big program, but we had to get started somewhere. Find your footing and determine for your organization if this is a priority. And recognize, like Dean, there are wonderful groups here that will happy to take your call and to help you and to guide you and to look at your nomination. Matthew and I have looked at a number of organizations' nominations just to kind of see where they are and cheer them on and help them out. So know that there's resources in your patient advocacy groups, and thank you, Dean, for bringing that up.
And one thing to add to that is you may think that there are some really big organizations who are in this space who are like, oh, they're too big, they're not going to help on this. That is not accurate. There are many groups who they're out here trying to build the infrastructure to support the work that you're doing. I'm thinking of one in particular, NBSTRN. I don't know if you all have heard of them. That's let's see if I remember, the Newborn Screening Translational Research Network. So their focus, and that is funded through NIH in a contract to the American College of Medical Genetics and Genomics. And so they really are working with data every day on a range of different conditions, and are happy to speak with anyone who's thinking about, newborn screening from a research perspective, and not just that early days of research, but really to translate it into public health, into clinical care. And so again, that's a really big group with a really big organization, but they're looking to help you all too, and thinking and being in conversation about what data should we be looking at? How do we count this? What does this look like? So I really encourage you to reach out to them. I know they've been, even if they're not necessarily a nominator of conditions, they've been very supportive to a number of different condition groups and patient advocacy groups through their journey in newborn screening. Do we have time for one more question? Okay, great.
I'm concerned about money. I know that everything is expensive, and both on the federal level and the state level. I'm concerned about the federal money not being there for newborn screening because they haven't reauthorized the money. And I'm concerned about the state labs where you have to help them find money to do the testing. I mean, I know you go to the legislature and say, we need more money. Is there other sources of money to help fund these things, both, I don't know, federally, but state-wise? Is there money to help?
Well, I'll go first. I don't have the exact answer for you, but I can tell you that the spaces you're speaking to are genuine, and you're right. Okay, so you're focusing very well in terms of what the issue is and where the money comes from. I can tell you from 2016 to today, we have, I think, 34 states that are now screening for MPS 1, which is approximately 76% of our population here in the United States. But what keeps me up at night, besides not having a therapy for my daughter, is the babies that aren't being screened in these other states because they don't have the funding. And I know this because these families are calling us, and now these children are 3, 4, 6, 8, and they've missed this huge opportunity with a life-saving therapy. And so newborn screening saves lives is crucial. The patient advocacy work around that area is crucial. And even today, right now, building your relationship with your legislators right now, going home and calling them this week and saying, we need this to get done. I can't get into political climates and things like that here, but I can share with you that it's important that your voice is heard, and heard in a strong way now. Even though we're going into that election process, we need to have that funding brought forward. Health labs, Scott Schoen, the director of North Carolina State Health Lab, gave us a really important lessons, and I've heard lessons over the many years of how they're so underfunded. And you have a voice there. We have a voice there that if you can partner with the health labs to be able to bring that potential advocacy component in your state forward, so that when you're going through the Office of Management Budget at the state level, they can consider different funding. But we have a lot of work, and we need newborn screening saves lives. And I'm grateful to Every Life, I need to say this, for looking at RUSP parity and looking at RUSP alignment, because this is absolutely one way that we're coming at it from a side, and we're just tackling it, like we're tackling it with RUSP alignment. So I'm so grateful for that.
The one thing I want to just add to that is, sometimes newborn screening is such an amazing program because it works so well. And unfortunately, we live in a world where people tend to focus on the crisis and the things that are completely broken, right? But we need to make sure as advocates that we're saying, no, we can continue to improve upon, and we need to be able to foster the work that's currently being done and to build upon it, and at the state and federal level. There are some people who think, oh, newborn screening, it's a state program. Why is there anything federal around it? Like, who needs money for that? So much of what we've talked about today is not, it's not a state's role to determine a federal, recommended uniform screening panel. It's not a state's individual role to, bring together all these experts and to do this work. And so I think taking that knowledge that you all are gaining from meetings like this and all the other meetings that you're a part of, and to say, no, this is what we mean by a system, and this is what it means to really fund a full system, was really critical. So with that, thank you so much.
Thank you.
Thank you. And we are going to break now for an hour-ish for lunch. Are there any other announcements about that? Oh, yes, I do. I know what that signal now means. So during lunch, we have a bit of an activity. So in this corner, we are setting up 2 boards, one where you can post the resources that you have, and if you have resources that you think would be helpful to the conversations we're having in this room and to other people who are thinking about newborn screening, you can write it down on a Post-it note and put it up. And if you're saying, gosh, I really wish there was this resource, I wish I could just type into Google, blah, 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 and then this would come up, put that up there. We want to see both what's out there and what the needs are, and see if we can kind of match some of that up. So that'll be an activity that'll be in this corner over here during the lunch period. And for those who are online, if you have resources you want to share or resources that you're looking for, please put that in the chat. Thanks so much.
Thank you.